Bismillahirrahmanirrahim. So friends, in this video we are going to talk about the mutagens. So let us start our video. Our topic for the day is the mutagens. So let us talk about mutagens. What are mutagens? So if we try to define the mutagens, these are the physical or the chemical agents that bring about the mutations. So these are the physical these are the physical or chemical agents that bring about mutation, that bring about mutation. So all those physical or the chemical agents that bring about mutation, that cause mutations of our genome, those are called as the mutagens. Now the mutagens, because they can be physical or chemical, therefore they are of two types. So these mutagens, they are of two types. Number one, there is the physical mutagens, and number second category of mutagens is the chemical mutagens. Number second is the chemical mutagens. Now physical mutagens, these include the radiations. Physical mutagens include the radiations, and in radiations, the pioneering work on radiations on X-rays was actually carried out by the Muller. What Muller did is that he studied the effect of X-rays on the Drosophila. So on the basis of his studies of X-ray on Drosophila, he is regarded as the father of actinology. So Muller is regarded as father of actinology because of his pioneering work on the Drosophila. Father of actinology. He is regarded as father of actinology. So now we will talk about the radiations. Radiations, as the physical mutagens, they are of two types. First category of radiations are the ionizing radiations. So radiations, they are of two types. First category is the ionizing radiations, and second category is that of the non-ionizing radiations. So we will here briefly try to differentiate between ionizing and non-ionizing radiations. So let us talk about the few points of differences between ionizing and non-ionizing radiations. So I will make two columns here. So let us make two columns here. So on one side, we will write the ionizing radiations. So here we will write ionizing radiations, and here we will write the non-ionizing radiations. Let us try to differentiate them. Let us try to find out few points of differences between them. Now, as far as the ionizing radiations are concerned, these are high energy radiations. So they have tremendous energy. These are high energy radiations. While as these non-ionizing radiations, these are low energy radiations. So they are low energy radiations. These are low energy radiations. As far as ionizing radiations are concerned, ionizing radiations, they can penetrate deep into the skin. They can penetrate deep into the skin, deep into the skin. So they can go beyond the dermis of the skin and can affect and can cause damage within the cells. While as these non-ionizing radiations, they can't go beyond the human skin. So these can't go beyond the skin. They are incapable of going beyond the skin. Those ionizing radiations, the examples of ionizing radiations, they include number one, X-rays. X-rays. They also include number second example, they include the beta rays and of course the gamma rays. Gamma rays. While as the example of the non-ionizing radiation include the ultraviolet radiations. So here are these include the ultraviolet rays. Okay. So let us now talk about how they bring about the mutations. Now ionizing radiations, they bring about mutations by the creation of the ions, free radicals, which directly damage our genome, which directly bring about changes in our genetic material. And these ultraviolet radiations, they are readily absorbed by our nitrogenous bases. So here I will be discussing about the effect of the ultraviolet rays on our DNA. So let us uh, discuss that on another slide. So what these ultraviolet radiations, or UV rays, they are readily absorbed. These are readily absorbed by the nitrogenous bases of our DNA. The nitrogenous bases, these include the purines and the pyrimidines. So these will be directly absorbed by purines and pyrimidines, and wherever there are present thymine residues adjacent on the DNA, at that they will result in formation of the thymine dimers. Suppose if this is the stretch of the DNA, suppose here is a stretch of the DNA, and we know that DNA is a double-stranded molecule having complementary DNA strands. So suppose here the nitrogenous bases present on one strand are just like this. It is adenine, and adjacent to adenine suppose is the thymine. Suppose adjacent to the thymine is present another thymine, then suppose another there is present then cytosine, and followed by guanine, followed by again thymine, and followed by again cytosine. Suppose this is the sequence. So we know that opposite sequence will be adenine is always base pairs with thymine, while as thymine will always base pair normally with the adenine. And here again there is thymine, so here will be again adenine. Here is cytosine always base pairs with the guanine. So suppose there is a guanine, here is guanine, and it will base pair with the cytosine. Here is thymine, and thymine always base pairs with the adenine. So here is the adenine, and again we have here cytosine, and cytosine we know it base pairs again with the guanine. Suppose this is the stretch of the DNA. So if ultraviolet rays are able to reach this DNA, then ultraviolet rays are readily absorbed by these purines, especially the thymine, and this results in formation of the intramolecular bond between the adjacent thymine residues, resulting in formation of thymine dimers. So these thymine dimers will be formed. Thymine dimers will be formed. Dimers will be formed, and these dimers won't be able to show the normal replication. So as a result of which DNA will get damaged, right? So this is the effect of the ultraviolet radiations on the formation of thymine dimer. So we can simply write down that ultraviolet radiations lead to the formation of thymine dimers. So wherever there are thymine dimers, so that portion won't be transcribed or not even be replicated properly. So this was about the radiation's physical mutagens. Now let us talk about the chemical mutagens. Now their chemical mutagens are of several types. We will again tabulate them in a table form, so that we are able to study them properly. So first we will talk about, so among chemical mutagens, we will first talk about the deaminating agents. There are certain chemicals which bring about deamination of the nitrogenous bases. Those chemicals are called as deaminating agents. So let us talk about the deaminating agents. Deaminating agent, one of the deaminating agent that acts as a chemical mutagen, that is the HNO2 or the nitrous acid. So that is HNO2 or nitrous acid. Nitrous acid is a chemical mutagen. What it does is, so let us write down what type of damage it causes in our DNA. So it results in the deamination of the cytosine, and we know if the cytosine residue gets deaminated, cytosine gets converted into uracil. So normally cytosine will base pair, normal cytosine will base pair with guanine, but once it gets changed into uracil, then it no longer base pairs with the guanine. This uracil, it base pairs with adenine. So instead of base pairing with the guanine, it now base pairs with the adenine. So what will that result in? So this results in the change of the bases. It results in the substitution of the bases. Suppose the normal base pair would have been adenine thymine, but this gets changed, or normal base pair would have been the guanine cytosine. Normal base pair was guanine cytosine. Here, as you can see, it was the normal base pair cytosine guanine, but now it gets changed into adenine thymine. Of course, here it is uracil, but in the next round of replication, it will be changed into adenine thymine. So here the guanine is being replaced by adenine, and you know, when one purine is replaced by another purine, that type of mutation is called as transition. So it is a typical example of transition. These deaminating agent like nitrous acid, it leads to transition type of point mutation. So transition type of point mutation, wherein one of the purine is being replaced by another. In this example, I have shown you that cytosine in this guanine is being replaced by the adenine. Here guanine, instead of guanine, adenine has been added. So therefore it is a type of the transition mutation. So this was the first chemical mutagen. Now let us talk about the another chemical mutagen. Another chemical mutagen includes second chemical mutagen. This includes the alkylating agents. Please pay attention. Alkylating agents. In case of the previous example, we had seen that deaminating agents, they bring about the deamination of nitrogenous bases. But here what we find is that this alkylating agents, they add alkyl groups to the nitrogenous bases and hence change their pairing properties. For example, examples of the chemicals which bring about the alkylation, they include the mustard gas, and you must know that mustard gas was the first chemical mutagen used, and it was actually used during the First World War. It was very much used during during World War II, sorry, and it was discovered by Sir Orbach. So mustard gas is one of the chemical mutagen which causes the alkylation, or it adds the alkyl groups to the nitrogenous bases, changing the pairing properties. Another example of alkylating agents, they include dimethyl sulfate, di, which is written as DMS. DMS stands for dimethyl, so I'm writing very slowly, dimethyl sulfate, dimethyl sulfate, right? And they also include, I am writing here, it also includes the DES. DES stands for diethyl sulfate. So what they do, these alkylating agents do is that they will add methyl or ethyl groups, which are the alkyl groups, to the nitrogenous bases. For example, in this case, then there is a possibility that if there is present guanine in the nitrogenous in the DNA, then after guanine gets ethylated, it becomes ethyl guanine. So normally guanine would base pair with the cytosine, but ethyl guanine is very unstable. Ethyl guanine, is very unstable. So when it is unstable, it is usually lost. So it is lost from the DNA. So unstable guanine, it is lost from the, it is lost from the DNA. It is lost from the DNA, and this results in a phenomena which is called as depurination. This phenomena is called as depurination. Depurination. So what is depurination? Depurination is the phenomena of losing purines from the DNA. So when purines are lost, we say that the DNA has undergone depurination, and this results in the deletion of a nitrogenous base, which can result in the type of mutation which is called as frame shift mutations. So alkylating agents, they can cause frame shift mutations because these alkylating substitutes of nitrogenous bases, their pairing properties are changed and they become unstable, and these unstable bases are later on lost from the DNA. So this was the second example of the chemical mutagens, that was the alkylating agents. So carrying forward, so moving ahead, so now in next slide I will be talking about now the further alkylating agents, chemical mutagens. Another example of chemical mutagens are, this is the third example, those are the base analogues. So let us talk about the base analogues. What are base analogues? Base analogues. Now base analogues, these are those molecules, those chemicals which show structural similarity with the normal bases. So these are the chemicals, which show structural resemblance with normal bases. So they will show structural resemblance with the normal bases. For example, I will cite an example, few examples rather. So instead of 5-bromouracil, 5-iodouracil, and 5-fluorouracil, these are the base analogues of nitrogenous base uracil. All these have resemblance, these resemble with the thymine. So instead of resembling with the uracil, these don't resemble with the uracil, rather they will be resembling, these resemble with the thymine. So they resemble with the thymine. 5-bromouracil, 5-iodouracil, and 5-fluorouracil, these are the examples of the base analogues which resemble with the nitrogenous base thymine. So instead of the thymine present in the DNA, sometimes these base analogues get incorporated. When base analogues get incorporated in place of the normal thymine, as in this case, this result is in the change of again the pairing, and the result is in the formation of either it results either in the deletion or in the substitution type of mutations. Similarly, one more example of this base analogues is another example. I will write in another with another color. So another example is 2-aminopurine. 2-aminopurines. So 2-aminopurines, we know purines are include the adenine and guanine. So aminopurines, be it adenine or guanine, they both resemble with the normal nitrogenous base, that is adenine. So again here, if instead of purine, adenine and guanine, the adenine nitrogenous base analog 2-aminopurine is incorporated in the DNA, that will again lead to transition type of the mutations. So this was about the third type of the chemical mutagen, that is the base analogues. So moving ahead, now we will talk about the another category of chemical mutagens, that is the hydroxylating agents. So this is the fourth type of chemical mutagen. These are called as hydroxylating agents. Hydroxylating agents. Hydroxylating agents. What are hydroxylating agents? Hydroxylating agents, these are those chemicals which will result in addition of the hydroxyl groups to the nitrogenous bases. Hydroxylating agents, they cause the hydroxylation of the nitrogenous bases. They cause hydroxylation, which means the addition of the hydroxyl groups. They will add hydroxyl groups to the nitrogenous bases. For example, if the base, that is cytosine, gets hydroxylated, it becomes hydroxycytosine. And this hydroxycytosine, I will write its name, this hydroxycytosine, I beg your pardon, this hydroxycytosine, instead of cytosine normally will base pair. Normally cytosine would have base paired with the guanine, but this hydroxycytosine, it instead base pairs with the adenine. Again, you can note here that guanine is being purine is being replaced by another purine, that is the adenine. So again, which type of substitution reaction is this? Substitution mutation is this? This is again the transition type of mutation. So again, this is an example of transition type of mutation. It leads to change of the bases. It does not lead to addition or deletion of bases, rather it leads to substitution of the base, and in this case purine, guanine is being replaced by adenine, which is again a purine. So such type of mutation is called as transition. So hydroxylations of nitrogenous bases, they cause the transition type of the mutations. And then finally we have the last category of the chemical mutagens, and in last category of chemical mutagens includes the dyes. There are certain chemical substances. This is the fifth one, probably, yes, fifth one, and these are the dyes. Now, dyes are the organic compounds having the cyclic structures. So these are the organic compounds having cyclic structures. These are the organic compounds having the cyclic structures. So these include, examples of these dyes include acridines. So this is the example of the dye, acridines. They also include the proflavin. So another example is proflavin. flavin. Now uh, what these dyes will do, what these dyes do is that they get incorporated within the DNA. Suppose here is the DNA molecule. Suppose this is the DNA molecule. This is the DNA molecule. Suppose this is the DNA molecule, and in this DNA molecule, suppose here is one nitrogenous base adenine, and opposite nitrogenous will be thymine. Then we have suppose cytosine here, and here it will be guanine. So these acridines, these dyes, they get incorporated in between the nitrogenous bases. Suppose they will get incorporated between this adenine and the cytosine, or between the adenine thymine base pair and cytosine guanine base pair. So once it gets incorporated within the DNA, they will result in the additions or deletion of the nitrogen bases. As a result of this intercalation, or as a result of their attachment within the nitrogenous base pairs of the DNA, that results in the addition or deletion of the nitrogenous bases. So as a result of these additions, nitrogenous base pairs will be either added or deleted, and we know whenever the nitrogenous bases are added or deleted, that results in changing the entire framework of the gene. So that type of mutations are called as frame shift mutations. So these dyes, they result in causing the frame shift mutations, which are also called as the gibberish mutations. So they result in production of the gibberish mutations. So here, gibberish mutations. Example of a disease which is caused because of frame shift mutation, that is the thalassemia. Thalassemia is one example of a mutation. Thalassemia, it is a blood disorder wherein abnormal hemoglobin is formed. This thalassemia is caused because of, it is E here, I suppose. Thalassemia is caused because of the frame shift mutations. So these were some of the examples of the mutagens. So I will very briefly recapitulate all of these slides before I end this lecture. So let us very quickly talk about what we have studied so far. So I will revisit all of the slides here. I suppose this was my first slide. So I talked about the ionizing and the non-ionizing radiations first. So ionizing radiations, these are the high energy radiations, and they can penetrate deep into the skin. Examples of these radiations include the X-rays, beta rays, and the gamma rays. While as non-ionizing radiations, they are low energy radiations. They can't go beyond the skin, and their examples of the non-ionizing radiations, they were the ultraviolet rays. Then we had seen how these ultraviolet rays, how they are able to cause the mutations. So these ultraviolet rays, we had seen that ultraviolet rays are readily absorbed by the skin, by the nitrogenous bases like purines and pyrimidines, and whenever there are present thymine pyrimidines adjacent in the DNA, they result in formation of the thymine dimers, as a result of which there will be addition or deletion of the nitrogenous bases. They won't be transcribed properly, and they won't be even replicated in a proper way, leading to frame shift or the gibberish type of the mutations. Then we had seen the chemical mutagens, and in chemical mutagens we had seen that chemical mutagens include Here are the chemical mutagens. They include the number first is the deaminating agents. Deaminating agent includes HNO2 or the nitrous acid, and this deaminating agent is they will remove the amine groups from the nitrogenous bases. For example, when cytosine gets deaminated, it is converted into the uracil, and then that uracil, instead of base pairing with guanine, it base pairs with the adenine, and this results in the transition type of the mutation. Sometimes, for example, if had there been guanine and that had been deaminated, then that would have resulted in formation of xanthine. It is also one possibility. Suppose if there were guanine here instead of the cytosine, then guanine after deamination gets converted into xanthine, and this xanthine also base pairs with the cytosine. So this type of mutation does not bring about any change or any substitution or any change in the nitrogenous bases. So such type of mutations are called as the silent mutations. So here is an example of the silent mutation, when the mutation does not bring about any change in the uh, type or the number of the amino acids. So this, for example, is the classical example of the silent mutation, where guanine after undergoing deamination gets converted into xanthine, but that xanthine also base pairs with the cytosine, which is the normal base to which guanine might have base paired. Then we have seen the alkylating agents, and alkylating agents, these include mustard gas, the first this chemical mutagen to be discovered by the Sir Orbach, and it was used during the World War II. And mustard gas, apart from mustard gas, we have dimethyl sulfate and diethyl sulfate. What they do is that they alkylate the nitrogenous bases. For example, guanine, after undergoing alkylation, ethylation particularly, it gets converted into ethyl guanine, and this ethyl guanine is unstable. It is lost from the DNA phenomena, which is called as depurination. So depurination, loss of nitrogenous bases, that leads to again gibberish mutations or the frame shift mutations. Then after this, we had talked about the base analogues. So base analogues, so base analogues base analogues, these are the chemicals which show structural resemblance with the normal bases. The, for example, 5-bromouracil, 5-iodouracil, and 5-fluorouracil, they have resemblance with the thymine, so they resemble with the thymine. And similarly, 2-aminopurines, they also resemble with the adenine, so they can lead to substitution type of the mutations. Then finally, last but one, we had talked about the hydroxylating agents. So as far as hydroxylating agents are concerned, yes, here hydroxylating agents are those which bring about the addition of the hydroxyl groups to the nitrogen bases. For example, guanine, the cytosine, when it gets hydroxylated, it is called as hydroxy cytosine. Cytosine, when it gets hydroxylated, it is called as hydroxycytosine. And this cytosine, normal cytosine would have base paired with the guanine, but this hydroxycytosine, it instead of base pairing with guanine, base pairs with adenine. Again, this leads to transition type of mutation. And then finally and lastly, we had the dyes, and in dyes we had seen that dyes are the organic compounds having cyclic structures, and these include acridines and proflavins. What acridines and proflavins do is that they get incorporated or intercalated in between the nitrogenous bases, leading to addition or deletion of the nitrogenous bases, which in turn leads to frame shift mutations or the gibberish mutations, for example the thalassemia. So I think this that is all for the day. Inshallah, tomorrow we will talk about the another topic. Till then, please take care of yourself. Allah Hafiz.